This video is a conversation with Dr. Nicolas Verhoeven of the YouTube channel Physionic about mitochondria and specifically mitochondrial dysfunction. Mitochondrial dysfunction is a common cellular feature of most chronic diseases. We see it in insulin-resistant liver and muscle cells and pancreatic beta cells that are unable to secrete enough insulin. And it therefore stands to reason that mitochondrial dysfunction is a key cellular process involved in the development of type 2 diabetes. But you can basically look up any chronic disease, from heart disease to cancer to neurodegenerative diseases. Mitochondrial dysfunction seems to be a common denominator playing a role in all of these conditions to some degree. So with Nic, I specifically focused on what we can do personally to keep our mitochondria healthy and happy. Enjoy. Nic, so welcome to Nourished by Science on YouTube. Really glad to have you here on the channel with us.
Yeah, sure. So first of all, it's an honor for me to be here. I've told you this off platform, but you are one of my top 3 sources of nutrition and science information. So it really is an honor for me to be here.
Maybe we'll start slow and get into, what are mitochondria, what's their basic function, and why are they important for our health?
Yeah, sure. So mitochondria are an organelle. So everybody, the viewer, listener probably knows that we have cells. Our body's made of billions and billions and billions of cells. And most of those cells, not all of them, but most of those cells have organelles. And the organelles are just these different structures within your cells that serve particular functions for the cell. You can think of them like, factories and different systems within the cell. And mitochondria are one of those systems. So they're one of these organelles. And in terms of their function, I guess we might as well get this one out of the way. It's the powerhouse of the cell. I guess we can go ahead and mention that. Close the podcast. I think we're done. So that's something that pops into people's minds often because it gets kind of drilled into us from like middle school into high school. And then of course, beyond that as well, that mitochondria are the powerhouse of the cell. So that means that they essentially generate cellular energy for the cell. Now, that said, they're important in more than just that one function, although that's what they're most well known for. And that's probably what they'll be well known for the rest of our biological existence. However, mitochondria have other functions as well. For example, cell signaling. So mitochondria have the ability to communicate with the other organelles and the enzymes and the different proteins, functional proteins that are found within the cell to allow the cell to function. So it's not just cellular energy that they generate. They also generate other compounds. They can sequester or take up different compounds, different molecules, different ions. They can create balance within the cell. They have a critical role in biosynthesis, so they can actually generate new molecules that can then be used for like hormonal production. So like cortisol, testosterone, estrogen, those are all dependent on mitochondria. And they also protect us from cancer. So there's a really powerful mechanism that when your cells become defunct and they ultimately start becoming cancers and start giving this rise to cancer, your cells have an independent mechanism to kill themselves, to essentially spare you from starting to develop cancer. But mitochondria also have this powerful system that works through different triggers, but ultimately leads to the same fate, that the cell ends up succumbing and dying off just so that it doesn't become cancer. So there's a whole host of different ways, and that's certainly not an all-exhaustive list for why mitochondria are important.
One question I hear a lot is people say, so they just make energy. So in other words, if my mitochondria don't work well, I feel like sluggish or weak. And they assume that is because, the mitochondria don't make enough energy for muscle movement, and there may be something to it. But the other thing certainly is that the energy, that the chemical form of energy produced in the mitochondria plays a major role in also providing energy for all other cellular processes, right? So all other transport mechanisms in the cell, enzymatic activities, just converting DNA to proteins, all of these processes require energy. And so a lack of functional mitochondria can lead to a deficiency in energy for all of these processes, right? Not just muscle contraction, which is what most people may be associated with energy that they need for the body. Now, an important term that people come across there is this term mitochondrial dysfunction. So basically, for some reason, the mitochondria stop working properly. Can you explain what we mean when we say, researcher may look at, say, the liver or a muscle cell and may say, oh, we observed signs of mitochondrial dysfunction. Like, what do they mean with this?
Yeah, so there's a number of different layers to this. So I'll go through 3 layers. The top layer is just mitochondria aren't functioning within a normal range. So we'll just say that's just kind of a real basic definition for mitochondrial dysfunction. A layer under that, which relates to how researchers might assess it, is they might look at particular markers of how mitochondria function. So we can actually go back to some of the ones that we were talking about in terms of the importance of mitochondria, because some of those can be quantified pretty easily. So we can look at, for example, oxygen consumption by mitochondria, because to generate energy, I won't go into the biochemistry, but to generate energy, mitochondria need to be taking up oxygen. So, that is the primary reason why we take up oxygen, is so that they can be delivered to mitochondria and then can go through this biochemical process that ultimately leads to cellular energy. So one way to measure is by looking at isolating mitochondria, and then, or even on a cellular scale, but usually we isolate mitochondria and then we look at their oxygen consumption rate. So that gives you one indication of their health. If it's high, well, that's typically a sign that, okay, it's functioning well, but then you probably need to look at some other indices as well, which we can certainly get into. Another metric that researchers might look at is ATP generation, which is a little bit more direct metric. Instead of just looking at oxygen consumption, they might look at ATP, which is, if we haven't mentioned it yet, ATP is the major cellular currency of the cell. The cell actually uses other currencies as well, other energy molecules, but ATP, or adenosine triphosphate, is the major one. So those are 2 ways that researchers can probe mitochondrial dysfunction. Then if we were to go to a 3rd layer, there's a lot of different ways that mitochondria can become dysfunctional that are not related to energy. So the loss of the inability to generate cellular energy is definitely one of them. However, mitochondrial dysfunction can come about due to a whole host of different reasons. So another one might be the loss of the membrane potential. So to explain that a little bit, your mitochondria have 2 membranes. They're kind of like this double insulated organelle. So there's an outer membrane and an inner membrane. And then on the very deepest inside recesses of the mitochondria, called the matrix, you have, in that region, it's much more negative than the outside of the mitochondria. And so we're still inside the cell, but the inside of the cell is more positive relative to the very deepest region of the inside of the mitochondria. Just keep in mind, the very deep inside part of the mitochondria is supposed to be very negative relative to the outside of the mitochondria. Now, if there's an imbalance, if you start to have more positivity on the inside of the mitochondria basically shuts off. I mean, there's no way for it to function anymore. And from that point on, it can actually typically start going into mitochondrial death. So it'll essentially get destroyed. And typically that goes by the process of autophagy. So autophagy of mitochondria, called mitophagy. Researchers get really creative with their names, which is great. So this defective or loss of a membrane potential. So the membrane potential is simply the difference between the inside of the mitochondria and the outside of the mitochondria. If you lose that, that is a sign of mitochondrial dysfunction. Defective mitophagy is another sign of mitochondrial dysfunction, where you might see a loss in the mitochondrial membrane potential, but it does not recruit the autophagy machinery. I won't go into all the different names of that, but the autophagy machinery would then come by and be primed. Oh, not that they're thinking about this, but biochemically, it gets targeted to a mitochondrion that's lost its membrane potential, and then it engulfs it and starts going through this process of destroying it. So you can get defective mitophagy as well. You can also get the accrual of different proteins on and inside the mitochondria. One example, our lab that I did my PhD in, we actually released a review where we looked at proteotoxicity. So it's essentially the overaccumulation of different proteins that are found on the outer membrane of the mitochondrion, but they can also find their way into the matrix of the mitochondrion. And if you have a bunch of dysfunctional proteins that are either not supposed to be there, they may be mistargeted. So instead of going to another region of the cell to fulfill the function that they're supposed to fulfill, they can get mistargeted to mitochondria and can actually attach to the mitochondrion. From that point, all the functional proteins that are on mitochondria may not be able to interact as well as they're supposed to. Or you may just get an overwhelming number of different proteins that just don't function anymore. So the accrual of these different proteins called proteotoxicity can lead to severe mitochondrial dysfunction as well. Physical damage, mitochondrial damage itself, which can occur through like excessive reactive oxygen species generation. So these are damaging molecules, or what can be damaging molecules, that can interact with different sections of the mitochondria and start ripping away at the kind of the chemical integrity of the mitochondria. So if you have excess levels of these molecules, then you can get damage to the mitochondria. I'm being very careful here to say excess amounts, because mitochondria do generate ROS, reactive oxygen species, for cell signaling as well. So it's not just that, you see a little bit of ROS and then therefore mitochondria are somehow abnormal. It's just if you get excess levels called oxidative stress. A lack of calcium buffering. I mean, I could keep going. There's a lot of different ways that mitochondria can become dysfunctional. I don't want to keep going and going and going. But if you want to know more, I can certainly cover more.
So no, I think this is actually really interesting discussion of all the things that can go wrong inside of the mitochondria and inside of the cell. The molecular things that can go wrong that can lead to mitochondrial dysfunction. And I'd be happy to chat more about this, but maybe the overarching question, if we take like 2-3 steps back and ask, what are the reasons why a person may develop mitochondrial dysfunction in the cells of certain tissues, or maybe all around the body? What do we know about the factors that may trigger mitochondrial dysfunction through maybe, and this is maybe how we can revisit some of these intracellular mechanisms, and through which intracellular mechanisms do we understand, if we do, how this happens?
Yeah, there's a lot of factors. I mean, some of them I mentioned, so like excess reactive oxygen species generation. Genetics is another one. Some people are born with mitochondrial dysfunction that can be kind of on a scale. So some people can have severe levels where they may not physically develop the way that they're supposed to, or you can have more minor versions which can manifest in particular situations. And I've spoken to people that have reached out to me that have been diagnosed with certain mitochondrial disorders, like MELAS is one, MELAS is one. And they're not extremely common, but when they do happen, they do arise specifically from genetics. And that's because you have like a loss of a particular gene, or you have a mutation in a particular gene that ultimately leads to the mitochondria not exactly functioning the way it's supposed to.
Can we stay there for a second? Would you mind describing what the phenotype is of some of these typical like genetic conditions that lead to mitochondrial dysfunction?
Yeah, so usually it very much depends on which mitochondrial disease you're talking about. But you can get, for example, an accumulation of fatty acids. So for the viewer, fatty acids, fat molecules, like the ones that you would consume from your nutrition or that would be released from your fat cells, they can accumulate within the cells that can ultimately lead to the cell having all kinds of different problems. And I know that you actually release a lot of content on insulin resistance. So that's a major way that it can present itself. Other ways it can ultimately lead to apoptosis. So that's cell death. So it's programmed cell death. So that can affect the organ systems, if that's like the liver, or if that's the brain, the eyes, all kinds of tissues. Any tissue that has mitochondria are going to be susceptible to these different conditions to varying degrees, because certain tissues rely on mitochondria more than other tissues. And there are certain ways of, where metabolism is very flexible. But again, we won't go down that road. In genetics, also Huntington's disease, if I could just quick mention like a disease that people might be aware of, or Alzheimer's disease, you can get an accumulation of dysfunctional proteins that again can accumulate on mitochondria. So for Huntington's, we know that there's a protein called Huntingtonin, I believe. It's kind of like an extension of the Huntington disease. That accumulates on mitochondria and stops mitochondria from being able to function like they're supposed to. Or with Alzheimer's, and just to be clear, I'm not saying that all disease is related to mitochondria. I'm also not saying that Alzheimer's all comes down to mitochondria. I think, if I can just go off on a side note just for a second, I think that when researchers get interviewed about their area of research, they start attributing everything to what they study, and I'm not saying that, but because there are confirmed mechanisms by which Alzheimer's is caused, but mitochondria, because of some of the same mechanisms, are related to these different disease states. So that's a few examples.
Okay, yeah, thank you. So maybe we could just take a look at where I interrupted you with the question: if is there something, say, that I could do or not do that affects the health of my mitochondria or could lead to mitochondrial dysfunction? And what do we know about those dietary and lifestyle factors, other exposures, and how they act through these mechanisms? So if we start, for example, with excessive reactive oxygen species accumulation, oxidative stress, like what do we know, what kind of exposures, lifestyle factors that we can potentially influence ourselves can lead to oxidative stress and as a result to mitochondrial dysfunction?
Yeah, sure. So there are I'll name two major ones, and then there are some kind of, I would call them like tier 2 factors that influence, that we can influence, that also influence reactive oxygen species generation, or over generation of reactive oxygen species. So actually, let me to really give the audience a clear picture of what we're talking about here. Let me kind of paint a picture of the structure of a particular section, a vital section of the mitochondrion, and then I'm going to attack both problems that I'm going to present in just a second through two things that people can do. So you have a mitochondria, and we've already described how it has two membranes. Now we're going to focus on the inner membrane. And in the inner membrane, there are these 5 proteins. Sometimes researchers just focus on the 4, but there's 5 proteins that are embedded, or 5 major proteins that are embedded into the inner membrane. And again, I'm not going to go through all the biochemistry of it, but The point being that you have substrate delivery at one point at one protein. And what I'm talking about substrate delivery, I'm talking about what comes from our nutrition, what comes from our fat cells, what comes from glucose in our bloodstream. And I'm going to go into that a little bit more in just a second. But just for now, just know that substrate for mitochondria to be able to produce energy gets delivered to this first protein. And then there's a chain reaction, which is why it's called, this chain of proteins is called the electron transport chain. There's a chain reaction that ultimately leads to the generation of energy. Now, if the cell doesn't need a whole lot of energy, but you have a whole lot of substrate, then one of the problems that can occur is you get what's known as electron slippage, which ultimately leads to the generation of these reactive oxygen species at a higher degree than what you'd normally want. Think about that scenario, and what are two things that you could do that could, A, reduce substrate, and B, create a metabolic sink that rapidly sucks up a lot of that ATP so that mitochondria do have a place to be able to generate this ATP. And one is dietary intake. And we can go into the specifics of that dietary intake, but the biggest factor is overconsumption. So if you are overconsuming in any way and you're starting to gain weight, that can lead to a lot of this excessive oxidative stress, which ultimately contributes to a whole host of different disease states. But on the far side, so sucking up ATP, the best way to suck up ATP is, we can focus on muscle. Certainly exercise is a massive metabolic sink, as in it just takes up so much ATP. As a matter of fact, I remember in one of my courses in my master's program, in one of my bioenergetics courses, the professor said that typically we consume or go through about 40 kilograms of ATP per day. But when you exercise, I don't know the exact amount of exercise. I think it was like a whole lot. It was like a marathon or something like that. You double that. So you go through 80 kilograms of ATP. So now imagine applying that to all your cells. That's such a huge demand for energy that it liberates, in a kind of a common term, it liberates mitochondria to be able to use up so much substrate and ultimately divert that towards ATP production, and far less of it gets diverted to reactive oxygen species production. Again, and just as a reminder for the viewer, it's not like the cells are thinking about this. This is merely just reactions happening. I'm sort of describing it like, mitochondria are deciding to put it one way or another, but that's not really what happens within the cell. It's a lot of reactions that are going on. So those are the two big ones. And then obviously we can go into more detail on some of the others.
So to clarify this for the viewer and connect it to some videos that I've made, when we overeat calories chronically, what happens is that the body tries to store those calories initially in the subcutaneous fat tissue that's all around the body, right? So everywhere we have subcutaneous fat tissue, and that's really the only really safe tissue to store fat long term. The problem is if we keep gaining weight, at some point, the subcutaneous fat tissue will be full. It will be unable to expand further and store more. And what happens then is that the body is forced to store any extra calories that come into the body in other tissues. And so the fat that we consume or the carbohydrates we consume that are being converted to fat, they eventually find their way into depots that are not primarily made for long-term fat storage, like the visceral fat depots in the abdominal cavity, so all around the inner organs, and also what we call ectopic fat storage. So that is fat storage in organs that are not at all made for fat storage. So good examples here are the musculature and organs like the pancreas or the liver specifically. And so what you're describing is that once we have reached the point where we're no longer able to store fat in the subcutaneous tissue, which is often referred to as crossing our personal fat threshold. So if we've crossed our personal fat threshold, we're no longer able to store fat effectively or efficiently in the subcutaneous fat tissue. Fat molecules start to accumulate in different tissues like the liver, like in muscle cells. And then they basically can become too much substrate, right, for this process that you're describing, if I understand you correctly. And that can lead to an impairment in the normal functioning of the mitochondria, such that it can lead to increased production of reactive oxygen species and what we commonly refer to as oxidative stress. Yes, describe that correctly.
Yeah, it was perfect.
I wanted to clarify this partly because my next question is directly linked to this, and that is, do we have any evidence to say that it is actually this way that it works, that basically we do start to accumulate, say, fat in muscle tissues? Let's just stay with muscle tissue for a second. We do start to accumulate fat in muscle tissue because the fat has nowhere else to go in the body, cannot be safely stored in the subcutaneous tissue anymore, but we keep eating, and so the fat finds its way into muscle cells and now accumulates there, and that is what causes the mitochondrial dysfunction and the increased excessive production of reactive oxygen species. Or do we have also evidence to suggest that the dysfunctional mitochondria could be there in the first place, and that contributes to an excessive accumulation of fat in the muscle cells? What's your view on this?
Probably depends on the disease state or the particular circumstance. Sometimes mitochondria may be affected by this overaccumulation of nutrients, and other times it's because you have these defunct mitochondria that you have this accumulation of nutrients. Let me actually talk about one study, though, that I remember reading years ago, where they took perfectly normal mitochondria, healthy mitochondria, and they applied, this is one of the points that I was going to talk about in relation to diet, they applied different types of fat. So the same level of fat, same amount of fat, but different types of fat. And they were able to show that for certain types of fat, which were unsaturated fats, there tended to be good outcomes for mitochondria, as in mitochondria would behave in a particular way. And what I mean by behave, the mitochondria have the ability to change their shape really drastically, from very linear and organized to extremely fragmented. But when they added a particular type of saturated fat, and I just want to be clear here that there are many different types of saturated fat for those listening, but the most prevalent, the most prominent is palmitate. So that's why they chose palmitate to apply to the cells. And that itself, as in mitochondria were perfectly normal, once they applied palmitate at the same level, again, suddenly mitochondria started massively producing oxidative stress. A lot of different stress enzymes were activated, and the cells ultimately were succumbed to that injury and ended up dying. So right there is at least some proof that it is the nutrition or the I guess I should say molecules that specifically affect mitochondria and cause this damage. But again, I don't want to say, oh, it's definitely that and it could never be anything else. I should be open to the possibility that could be reversed as well.
I actually really liked what you said there, that you felt that there is good evidence to suggest that, even someone who has perfectly good mitochondria, let's say in the musculature or in the liver, that if they cross the personal fat threshold and they're unable to store fat safely in their subcutaneous depots, the fat finds its way into other tissues. So suddenly fatty acids start to arrive in muscle cells, they start to arrive in liver cells, that accumulation of excessive substrate, right? Excessive fatty acids, maybe also excessive glucose, can in and of itself lead to mitochondrial dysfunction, but that it could also be that other factors cause the mitochondrial dysfunction, and that contributes or at least exacerbates the accumulation of fat in the cell, right? Is that do I summarize that correctly?
Yeah, definitely.
So in this context, I would actually like to stick with this topic a little bit because it's very fascinating in terms of one thing we haven't really talked about, and that is tissue specificity. And that what I mean with this is that, let's say if we look at a fat cell very obviously is well equipped to handle a lot of fat, right? So a subcutaneous fat cell, for anyone who's ever looked at these under a microscope, you have, let's say another cell type, let's say a macrophage, a cell type of the immune system. It's like a tiny cell. And then a fat cell would, by comparison, have this type of size, right? So they're huge compared to most other cell types. And they're filled, seemingly till they could burst with just one huge fat droplet, in the form of triglycerides, similar to the form of fat, say, in butter. So very obviously, it does seem that in the fat cell, this huge accumulation of fat doesn't lead to mitochondrial dysfunction, but much smaller amounts of fat, say in a muscle cell or liver cell, can wreak a lot of havoc, including on their mitochondria. Can you comment on that?
Yeah, actually, I've got a great example of mitochondria's relationship to fat in muscle that is quite different from, well, actually ties into some of the things that you were saying earlier about ectopic fat as well. So there are examples of lipid droplets, so these kind of sacs containing all these different fats within them in muscle as well. And for highly trained individuals, so people who exercise quite a lot, there actually starts to develop some of these lipid droplets within the musculature. However, the relationship to mitochondria is fascinating because the placement of these lipid droplets is what's known as myofibrillar. So I'm not going to go too into depth with that because I don't want to lose people with some of the technicalities. But the point being that it's kind of in the deeper recesses of the cell, and the location of mitochondria are right next to the lipid droplets. And the belief is that the reason why this happens is so that the lipid droplets can deliver, or somehow that these fatty acids and these fat molecules can be delivered to mitochondria in such close proximity. On the other hand, for individuals who are severely diabetic, people who might be obese, they tend to have in the same exact tissue, so muscle tissue, muscle cells, they can also have lipid droplets that start to accumulate, which is kind of relating to this ectopic fat, but they tend to be on the surface of the cell and tend to be related to far more insulin resistance. So the exact same phenomenon happens, but simply because of the location of the fat and the relationship to mitochondria, we're seeing completely different scenarios between those two conditions, which when I first ran across that research, I just found it absolutely fascinating that you can have that difference. And then once you start speaking about like fat cells, to your point about this one massive lipid droplet and the relationship, I mean, there's just so much complexity there of, well, why aren't mitochondria in those fat cells? And of course, you get into fat cell subtypes like beiging of fat and the brown fat. And brown fat obviously has this tremendous relationship with mitochondria that white fat doesn't. And it gets so complex. But the point being that there's a lot of discovery to be had there in terms of why mitochondria react certain ways to the context of the cell that they're in and the context of the fat that's around the mitochondria.
And so is it your impression that, say, in the case of an endurance runner, this is a carefully orchestrated process where the cell actually notices, repeatedly we have demand for a lot of energy, and so we'll just create a planned store of a little fat droplet inside the cell next to the mitochondria? Whereas in someone who's developing insulin resistance and diabetes, who just overconsumes calories, it's more of an accidental deposition that just happens and that the cell is forced to deal with, whether it wants it or not. Is that maybe a fair way to kind of think about this?
Yeah, that's how I think about it. I mean, obviously, you'd have to talk to a researcher that specifically studies exactly those phenomenon. But my impression is that's exactly right. That although a marathon runner or an endurance athlete, they typically don't have a ton of fat on them, and what fat is there that gets released from the fat cells has a long way to go just to get to the musculature. So the cells end up developing these lipid droplets that are in extremely close proximity with mitochondria. On the flip side, with individuals who suffer from obesity, the fat depots, I don't know if you've looked at some of the literature on the exchange of fat in the fat cells or and around the fat cells, but there's this massive exchange that's like way above normal, where it's almost like the fat cell simply can't hold on to the fat and just starts like regurgitating it back out. And there's some fascinating literature with immune cells as well, where immune cells will start to crowd around the fat cells and will start taking up the fat. Now, what they're doing with that fat, I don't know. But my hypothesis, not that I'm ever going to test this, but my hypothesis is that the immune cells are trying to help oxidize some of those fats, which, if that turns out to be true, would be incredibly cool, this symbiotic relationship. But the point being that with people that suffer from obesity, they tend to have higher triglyceride levels in their bloodstream, which tends to then, it could accumulate into the musculature and all these other lean tissues. And I wouldn't be surprised if that happened in other lean tissues as well. It doesn't necessarily have to be muscle, but that's at least what I've looked up.
Yeah. So let's maybe think about the second part of the equation that you went into, and that is the use of the ATP. So if let's say we have a muscle, there's some fat accumulating there, and we start to exercise, even if just going for a long walk every day, right? What happens is the musculature needs more, these muscle cells need to generate more ATP. And so what if I understood you correctly, one of the positive effects of that is that it just starts to burn up more of the accumulated substrate, like the glucose, the fatty acids, in a way that is less likely to lead to excessive reactive oxygen species generation. Yes, is that proper summary?
Yes, but there's some additional benefits of exercise that are not found from the previous part of that equation, looking at nutrition, or at least not as heavily, I should say. Exercise also has this potent effect of, because it's so demanding on the energy supply. Just as a quick biochemistry lesson, if you want to call it, ATP gets changed. I'm just going to use kind of general terms, but changed to ADP, so adenosine diphosphate. So triphosphate means that it's got 3 phosphates, then diphosphate, 2 phosphates, and then it can actually be further converted into AMP, so monophosphate, so a single phosphate. That dramatic drop from ATP to ADP, and then even from ADP to AMP, is a massive cellular signal, which ultimately affects a protein that a lot of people are pretty familiar with at this point, because there's a lot of hubbub about autophagy, which is AMPK. AMPK is this protein that it can actually be bound. The reason why it's called AMPK is because AMP, these molecules that start to build up due to exercise, because you're just using up such high amounts of ATP and producing ADP, which again can get converted to AMP, although it can also be turned back into ATP, but that's a different story. The point being that as AMP levels rise and ADP levels rise, that can activate AMPK. AMPK then has a tremendous impact on another protein called PGC1 alpha. And PGC1 alpha, and I promise I'll stop spitting out different protein names, but the point being that PGC1 alpha is a mitochondrial biogenesis factor. It's the most potent one. So this protein, which is highly stimulated from exercise, can then activate mitochondria to start generating more mitochondria. So because you're essentially creating this quick stressor, and I mean quick by 30 minutes, an hour, a few hours of exercise relative to your 24-hour day, that quick exposure to this stress of a sudden drop in ATP and a significant increase in ADP and AMP leads to mitochondria, or the cell, starting to produce more mitochondria, which ultimately bolsters the ability for the cell to then be able to produce that ATP and handle the next bout better, and then the next bout better, and so on and so forth. So it has this additive effect that exercise has that nutrition doesn't quite have.
Mm-hmm. Yeah, thank you for that. So to recap this part is one thing that is I'd say pretty clear is that if people chronically overconsume calories, once they cross their personal fat threshold, the glucose, the fatty acids accumulate in other tissues, like the muscle cells, the pancreas cells, the cells of the liver, probably others. The brain is, I think, a hot candidate for this as well, right? So there is then this consequence that the mitochondria cannot quickly enough metabolize all of this fat, which immediately has been linked to insulin resistance in these tissues. But in addition to that, the increased availability of substrate in the cell seems to contribute to mitochondrial dysfunction, partly through an increase in excessive reactive oxygen species generation. So the conclusion there obviously would be try to eat in a way that doesn't lead to chronic overconsumption of calories, right? So I think that's probably one of the most important things anyone can do for their health, is to eat in a way that it doesn't exceed their total energy expenditure. And ideally, if they have excessive fat beyond their personal fat threshold, they should do whatever it takes to get rid of that. The second part is exercise, which increases demand for ATP through the mitochondria and also makes the mitochondria run more smooth, simply because it creates a tremendous pull for ATP in a way that also reduces excessive reactive oxygen species generation and basically contributes to keeping the existing mitochondria healthy, while also providing a stimulus through the use of ATP to generate new mitochondria. So in other words, if you exercise regularly, the number of mitochondria in your muscle cells is going to increase, right? Is that correct?
Yeah, absolutely. Yeah. And it'll increase tremendously, actually.
So if we close that loop for a second, because you started at the beginning by saying that in a cell culture dish, you can test the function of your mitochondria and detect potential dysfunction by looking at the rate of oxygen consumption. We have a test like this for the whole human, right? The whole organismal level, where you can go into a lab and measure your VO2 max. Is that the right way to think about this? That if we regularly keep our, have lifestyles that keep our mitochondria healthy and exercise such that we actually increase the number of mitochondria, that is the mechanism through which we also increase our VO2 max?
Yeah, exactly. So I think of VO2 max as kind of this human proxy for mitochondria, but I want to be clear that it doesn't necessarily just represent mitochondria, because you could have a low VO2 max that is not because of defunct mitochondria, but because of something else. You may have a pulmonary issue, you may have a cardiovascular issue, or something along those lines. So I don't want to say that it's a 1-to-1 ratio. VO2 max immediately talks to mitochondria, but assuming everything else is relatively healthy, it does. I mean, you're literally consuming oxygen so that it can be delivered to red blood cells, so that it can be delivered to all these tissues, so that can then be pushed into mitochondria for energy generation. So absolutely, there's a clear relationship there.
Yes. Yeah, thank you for clarifying that. I think it's really fascinating to think about it this way, right? So if you have a high VO2 max, that simply means your entire system works well, right? Your respiratory system works well to get the oxygen into you and get rid of the CO2. Your cardiovascular system works well to transport the oxygen around the body, and your musculature works well in using the oxygen. And that is what happens in the mitochondria to generate the energy that's needed for the exercise. So let's maybe move on where, we've discussed that excessive accumulation of substrate in the cell or too low a demand because we're just sedentary all the time can lead to mitochondrial dysfunction. Now, there's several other factors that I think have been linked to mitochondrial health. And I wanted to get your opinion on some of those. So, for example, in my last two videos, I spoke about light and this whole new area of near-infrared light potentially acting on mitochondria. And I also spoke about cacao, dark chocolate, as a source of polyphenols that seem to have potentially antioxidative properties that may be linked to improved mitochondrial function. And there's certainly many other examples like that, where certain exposures we may have, good or bad, could either help us keep our mitochondria working properly, or that in and of themselves could lead to mitochondrial dysfunction. You've mentioned earlier already certain fatty acids, like palmitic acid, if they accumulate within the cell, they might by themselves contribute to mitochondrial dysfunction. Can you think of, what do you think, first of all, of like, how do you think about these types of factors in the grand scheme of things? And do you have other examples?
Yeah, so I was going to bring up some of these. I hadn't intended to bring up the red light or near infrared light, but it is an area that I've looked at as well. I'm always a little dubious about those studies because it's incredibly difficult to have proper controls for that kind of study. But putting that aside, there are a number of studies that have shown that the effect that red light or near infrared light has on mitochondria and does seem to improve the efficiency of mitochondria. I think the mechanisms are still a little bit being worked out. I know that it affects some of those 5 proteins that I mentioned in the inner membrane. It seems to directly affect some of those. However, also there's something called retrograde signaling from mitochondria. So just like I was talking about with the changes in ATP level from exercise, that is sensed by the entire cell. It's not just that it affects AMPK, it affects all kinds of different aspects of the cell. So red light seems to have an effect on that kind of signaling. So the exchange of these different molecules within the cell. And in terms of polyphenols and really just fruits and vegetables and anything that's abundant in antioxidants, that tends to be a benefit for mitochondria, more specifically if other aspects are leading to, let's say, more oxidative stress. Then these antioxidants can come in and affect mitochondria and can kind of quiet, almost think of it like a burn, can quiet the burn that's occurring due to this oxidative stress. I will say also exercise, just as speaking to exercise, another benefit of exercise is that it can stimulate mitochondria to stimulate the production of internal antioxidant systems as well through these reactive oxygen species. In terms of other things that come to mind, I had exercise, gaining weight, as in you would want to avoid that. An antioxidant-rich diet is something that a person would want. And then there's a reduction in palmitate-based saturated fats. That was another one. And also something that I don't think I've ever heard anybody else talking about this. Not that this is like some massive revelation, but taking care of other systems within your body. What I'm talking about is your cardiovascular system. Something that can damage mitochondria is the excess of oxygen or the lack of oxygen in particular situations. So when we're talking about like atherosclerosis, for example, when there's a coronary artery that gets occluded, that gets blocked up, and you have a lack of blood flow, which means that you also have a lack of oxygen delivery to downstream areas of the heart, those cells have a sudden drop in oxygen delivery, which means that mitochondria have no way of producing energy. However, one thing that we found in the research literature is that if you suddenly open that occlusion and just allow all the blood flow back through that area, that can lead to what's known as reperfusion injury. And one of the main reasons is that mitochondria get suddenly flooded with all these oxygen molecules that ultimately lead to a mass spike in reactive oxygen species. So I know that's an incredibly specific scenario, but the overall takeaway there is that improving your atherosclerosis risk and just making sure that you overall try to avoid those kinds of different coronary issues is a great way to also maintain mitochondrial health. And it just turns out that a lot of the things that you would do for that also just directly affect mitochondria and improve mitochondrial health.
So one thing that I was wondering about is if we develop mitochondrial dysfunction in different diseases, like if we look at the literature, right, we'll find evidence, say, in pancreatic beta cells, for example, that are unable to produce enough insulin. We'll find evidence there of mitochondrial dysfunction. In insulin-resistant liver cells and muscle cells, we'll find evidence of mitochondrial dysfunction. But also in, let's say, certain brain regions, in neurodegenerative diseases, we may find evidence of mitochondrial dysfunction. And so the question is, are all sorts of mitochondrial dysfunction the same, or it just matters which tissue is affected and then that tissue basically gets sick? Or do we have good understanding of like tissue specific types of mitochondrial dysfunction? Like, how can we think about that?
Yeah, and this is educated speculation, just to be clear, because I have not looked at I haven't related the different types of mitochondrial dysfunction to specific tissues or specific disease states, because it's usually talked about just kind of as a overall definition. Mitochondrial dysfunction is related to Alzheimer's, diabetes, cancer, all these different aspects. But my intuition is that no, they are very different. From disease state to disease state, but also when you're talking about tissues as well. And I think the best evidence of that would probably come from some of the genetic studies that have been done. If you're looking, I can go back to the Huntington example. That's a particular type of problem or mitochondrial dysfunction that's related to the overaccumulation of those proteins. Meanwhile, if you were to look at obesity, it's probably not necessarily, or not in most cases, genetic related in terms of the mitochondrial dysfunction, but more so the overabundance of the nutrients, the substrate that's available, or being sedentary would be another one. That's insulin resistance is also improved by exercise. So the lack of exercise can also be another way that you get this dysfunction, because you have more reactive oxygen species, for example, or you have fewer mitochondria, you have fewer healthy mitochondria, you have mitophagy isn't as functional. So I would argue, if it were just me kind of speculating on this without looking at the research in depth, I would say that's probably different from one tissue to another tissue.
So if the viewers now have become curious about their mitochondria, mitochondrial dysfunction, like, is there any test they could have done to figure out if they have mitochondrial dysfunction? Or if not, are there certain indications that people could use to at least get a sense of what the state of their mitochondria is?
So there are tests. Some of them are better than others. You'll have some companies that will do tests where they just kind of measure the overall amount of mitochondrial proteins, like the 5 that I mentioned in the inner membrane. I do not consider that to be a good test of mitochondrial function or dysfunction. It tells you that you have a certain number of these proteins, but it doesn't really tell you anything else. However, I know the Mayo Clinic, this is not something that's like readily available. You'd have to actually get a prescription or a consult with a physician that would then say, okay, yeah, I've got you slotted with the Mayo Clinic. And then they do a huge gene array where they specifically target these different mitochondrial genes that they know are highly related to mitochondrial diseases. And they can also look and see if some of those are mutated, and then that would give you an indication. Beyond that, I should also say that, and going back to your previous point about the different tissues, it does depend on the tissue. Like, you can get a sample from, let's say, your skin or fibroblasts or even muscle, but that doesn't necessarily speak to the entire population of cells in your body. And that's always been the case. If we kind of put that argument aside, I don't think that there are any like really readily available ways to measure mitochondrial function or dysfunction. Aside from you know, we did mention VO2, so that is one way, but again, that's not a one-to-one relationship. Beyond that, I mean, if you want to go to a lab, if you want to volunteer, you can always volunteer, and maybe some researcher will do it for you. But I in general, it's just a very difficult thing to measure, just because there's so many different variables that you have to take into account.
Yeah, I actually think this is also something that holds research back, actually, right? Because people who do intervention research, let's say, I used to do clinical intervention trials with different dietary exposures. And, we didn't measure mitochondrial function in muscle or liver as an endpoint because it's very hard to measure, right? So, and that's partly why, the evidence we have in terms of what the actual cause-effect relationship is, like what happens first, for example, what comes next, is at this point suboptimal, right? It's inconclusive because we simply haven't done the longitudinal studies with different interventions where we know for sure, okay, well, this is the status of the mitochondria now, and then we eat, whatever, chocolate or broccoli or eggs, and this is what happens then to them. So I think that's to some degree, a reason why some of this research is based, say, on animal experiments, on cross-sectional studies, on cell culture work, and so forth. And there's certainly a lot still to learn, but I do hope that it has become clear in this conversation to people that we do already know quite a bit about it. The mitochondria are undoubtedly very important for our normal function of all of our cells, which means our entire body. And the factors that we know about for sure that keep our mitochondria healthy are generally the factors that we also understand are important for us to be healthy, feel good, and minimize our risk of chronic disease, such as overconsumption of calories. Chronic overconsumption of calories is definitely a very bad thing for our mitochondrial health. Lack of physical activity, so chronic sedentary behavior and just never really exercising is a second very bad factor for your mitochondria. And a diet that's low in antioxidants. So, if we frame this differently, we would say the three things you for sure can do to keep your mitochondria healthy is make sure you eat a healthy diet that is rich in fruits and vegetables and other antioxidant-rich foods that helps you eat to satiety without over-consuming calories. And just think of yourself as someone who needs to move regularly, be that in formal exercise sessions like going for a run or doing resistance training, but also just in terms of minimizing sedentary time, right? Where we know for sure that if we sit for 4 hours at a time, that's not a good thing. And so maybe breaking it up and going for a walk in between or just doing a few squats. All of those are almost certainly things that will be beneficial for your mitochondria and as a result for your metabolic and whole body health in the long term, right? Well, Nic, I really appreciated you taking the time for this conversation. I thought this was very interesting. And where can people find you, learn more about you, and connect with you?
Well, it was a ton of fun for me too, considering I got to talk most of the time. So anytime a scientist gets to talk about what they love to talk about, no complaints there. Also lovely company. But people can find me at Physionic, so PhysioniC. So it's physio, like physiotherapist, but just cut the therapist and put NIC in there, stick them together, and you got me. I'll show up on Google. I'll show up on all the AI models. I'll show up everywhere. So just look that up.
Yeah, and I'll leave a link to the excellent Physionics channel in the description box below this video.
Thanks.
So thanks so much again for this conversation, Nic.
Yeah, thank you.
That was my conversation with Dr. Nicolas Verhoeven of the YouTube channel Physionic. I have left a link to his channel in the description box below, and I very much recommend that you check it out, because Physionic is one of the few nutrition and health channels on YouTube that is strictly evidence-based and that does not engage in sponsorship deals or affiliate marketing. That is the same approach that I've chosen for Nourished by Science, and if that is something you appreciate, consider becoming a patron of the channel on patreon.com. Being a patron comes with some perks, such as being able to suggest future content, asking me questions about published videos, and even letting me know what you'd like me to include in a video while I'm working on it. I also offer regular live video calls in which you can directly discuss the science with me. I've left a link to our Patreon page in the description box below. Lastly, don't forget to please the YouTube gods by pressing the like button below if you in fact liked the video, and let me know in the comments if you have any questions. Thank you for watching. Take care.